Okay, for this next lecture, we're going to talk about cell signaling. And so basically, for cell signaling is one of the ways that a cell can respond to its environment. It's one of the ways that your body can tell cells or organs made up of cells what is needed and what needs to happen. So cell signaling really brings together the cause and effect. So there's something out there in your body that is telling your cell what to do, and the cell performs some function in response to that. So the general features of cell signaling are pretty straightforward. You basically are going to start with some sort of signal, and so the signals are going to be by messengers. The messengers can be listed here. We have hormones, insulin, glucagon, we have epinephrine. We're going to have neurotransmitters, acetylcholine, for example. Cytokines are going to be messengers of your immune system. So cytokines are immune messengers. And then we have our retinoids as well as growth factors. Growth factors are going to be involved in many processes, including things like hematopoiesis, your making of your blood cells. Different growth factors are going to promote the growth of different cell types. Now, we also are going to have some of our messengers bind to intracellular receptors. So a nuclear hormone is going to bind intracellular receptors. And then these intracellular receptors with the hormone attached are often, or more often than not, become transcription factors. And transcription factors are just elements that can bind to regions on the gene and turn on some sort of gene. So our signal transduction, again, is going to sometimes end on a point where you're going to turn on genes. And then you can also have some chemical messengers such as nuclear hormone messengers. Vitamin D is such a messenger. They have intracellular receptors that also can go and turn on genes. Now, there are also going to be hormones or chemical messengers that bind to a receptor on the surface. And these receptors are the specificity. These receptors are very specific to the hormone. And so the receptors are going to be involved in transducing a signal once they're engaged with their specific hormone. Some types of surface receptors are going to be ion channels. We also have tyrosine kinase receptors. An example of this is insulin. Insulin uses tyrosine kinases, glucagon, and epinephrine uses the G protein coupled receptors. So regardless, chemical messengers' purpose is to elicit a response in a target cell, and they themselves are not metabolized. Now, there are some terminology associated with signaling and these chemical messengers. So we have in our endocrine system that they can release a chemical messenger, and these can work on a distant target. An example of an endocrine messenger is estrogen. There also is paracrine release of hormones. So that can have a local target. So an example of a local target is acetylcholine, which is going to be released by a nerve cell, and then is going to act on a target cell, such as a muscle cell. And then finally, we have autocrine. Auto means self, and so these can make the hormone, and then the hormone can have a specific receptor that it binds to on the same cell that made it. So in this case, and the immune system uses this a lot, so this can be a cytokine. And the cytokine can be released by your immune cell, and then it'll bind to a receptor on the immune cell and transduce a signal inside that immune cell to do something. Now, there are three major chemical or systems that use chemical messengers. So we have our nervous system, which is going to have two types of messengers. They can have small neurotransmitters. And they can also, and these are also referred to as biogenic amines. They're also going to have neuropeptides. And so you know what a peptide is. A peptide is a primary structure of a protein. So that's exactly what a neuropeptide is. An example of some neuropeptides are going to be acetylcholine, epinephrine, GABA, for example. All of those are going to be neuropeptides. The next system is the endocrine system. The endocrine system is, of course, going to have hormones that are going to be made by endocrine cells that are a part of endocrine glands. And these are going to transport it through the blood through distal sites in the body. Some examples of these can be polypeptide hormones such as insulin, and the polypeptide, of course, just means more than one peptide, and peptide, again, is that primary structure. You can have steroid hormones. And steroid hormones are going to be derived from cholesterol. And then you can also have thyroid hormones, which is going to be derived from tyrosine. And the catecholamines, which, for example, is epinephrine. Now, keep in mind that epinephrine is part of the endocrine system, so it can get in the blood and it can travel. But it is also referred to as a neurotransmitter, so it can act locally as well. Now, not derived from endocrine glands, but these are also classified as hormones, are retinoids, and which are vitamin D A and are vitamin D. Now, the last system that uses chemical messengers is our immune system. Immune systems are going to use cytokines, and these are small chemicals, small proteins that are going to communicate with the immune cells. And so some classifications of these are our interleukins, which are IL, and we also have tumor necrosis factor. This is also TNF. We also have interferons. These are antiviral. We have chemokines. And chemokines are sort of addressins. They tell the immune cells where they need to go. They tell them where to travel to. They tell them where they need to hang out. They give them basically an address that's part of the body. So there are other chemical messengers as well in the immune system. and we'll go over them more in biochemistry too. We have eicosanoids, so we're going to have a whole lecture on our eicosanoids, which are going to be our prostaglandins. There are growth factors, as I mentioned before. There are platelet-derived growth factor, and so when we talk about hematopoiesis, we will bring up the different growth factors. Now, as I mentioned before, if a hormone is going to bind to an intracellular receptor, the hormone and receptor become basically a transcription factor. So here in this case, your hormones are going to be delivered on what's called a carrier molecule. So some carrier molecules are very specific for a hormone, such as your thyroid hormone is going to have a specific carrier molecule. Others are just going to travel with serum albumin or some other protein that's going to carry the hormone to its target. Now, on the intracellular receptors, these are going to cross the membrane, shown here, bind to a cytosolic receptor, this little kidney bean showing thing, and then the hormone and the cytosolic receptor are going to move into the nucleus, and they are going to turn on some set of genes. You can also have a hormone that's released that gets into the nucleus and it has a specific nuclear receptor. So either way, they're both able to cross the membrane. Both triangles can cross the membrane, triangle and circle, and then they can interact with their specific receptors either in the cytosol or in the nucleus. And these are going to be lipophilic hormones. So they love the lipid membrane, and they're going to cross the lipid membrane and then bind to the receptors. Now, most of these, again, are going to interact with the DNA and turn on genes, and the genes they turn on are dependent on the signal from the actual hormone. So what is the hormone trying to tell the cell to do? And then we have our hormones that are hydrophilic, love water, and those are going to bind to the cell surface. So these are our cell surface hormones. They're going to be hydrophilic. So they don't cross the membrane. They're going to be specific for a specific receptor, and these can be peptide hormones, cytokines, or catecholamines, and they are also going to transduce a signal inside of the cell through that receptor. Now we also have our nuclear hormone superfamily. These are our lipophilic hormones. Oops. And so one example is a thyroid hormone superfamily. So steroid hormones, thyroid hormones, retinoic acid, which is vitamin A, vitamin D, are all going to have the ability to cross the membrane directly, and they all are going to bind to the receptors and become transcription factors. So all of these listed here can do that. Most are going to be transported in the blood and associated with serum albumin. Serum albumin is made by the liver, and it's one of the most dominant or most abundant proteins in your actual blood. And it's because the liver is constantly making it so that anything that is hydrophobic, such as a steroid hormone, is able to be transported through the blood bound to serum albumin. Now, the receptors are going to be steroid hormone receptors, thyroid hormone receptors, and most of these receptors for this particular class of hormones, the lipophilic hormones, are going to be in the nucleus. Now, the plasma membrane will have receptors for some hormones. So some examples are an ion channel. This is for our nicotinic acetylcholine receptor. We also have our G protein receptors. We've talked about G proteins. G proteins are going to transduce signals through our cyclic AMP that we've talked about already, pKa. We have our receptor tyrosine kinase. That's going to be our insulin receptor. We'll have a discussion about that. We have adhesion receptors, etc. So there's a lot of different receptors. Now, one of the things about plasma membrane receptors is they have membrane spanning alpha helixes. And their extracellular domain is going to bind to the membrane or to the messenger, and the intracellular domain initiates the cascade, the signal cascade, which will amplify whatever that messenger is trying to tell the cell. They're going to have two types of responses. The first response is fast, and this kind of response is going to be very rapid. It's associated with ion channels or enzyme activity. Let ions in, ions out. Phosphorylate some enzymes, dephosphorylate other enzymes. Oops. And the second is going to be slow. And this has to do with gene expression. So gene expression will be slow on, slow off. The rapid response will be quick in, out for ion channels, or quick on, off for enzyme activity. Now there are 5 features of signal transduction. There's going to be specificity, so you have this signal molecule is going to fit to the binding site of its receptor. Now the specificity is such that other hormones are not going to be able to bind to that receptor. So insulin binds to insulin receptor, epinephrine binds to epinephrine receptor, etc. Affinity is going to be the degree to which a substance binds another receptor or its receptor. And so you're going to have different, some receptors require cooperativity, other receptors have controls over binding. But it all goes back to specificity. The hormone has to be able to bind to the receptor to start the signal. And then you have something called amplification. And amplification is going to be basically the enzymes are going to activate an internal signal cascade made up of enzymes. Or hormone, I'm actually going to say messenger. Messenger activates and these enzymes and the number of molecule increases as the enzyme cascade continues. And so as an example, you have your signal, and that's 1, and this is going to activate enzyme 1. And then enzyme 1 will activate enzyme 2, but more than 1. And then enzyme 2 is going to activate enzyme 3. and so on and so forth. So this activates enzyme 3, multiple 3s, and so you just get amplification of the signal as you go through. So now 3 can activate 4s, and 4s can activate 5s, and so you get this amplification starting with just a single signal. And then there's also a concept of modularity in which proteins with multivalent affinities form very diverse signaling complexes. Phosphorylation, for example, provides reversible points of interaction with an enzyme, so that's modularity. So one way to think about this is allosteric regulation. Different groups combine to enzymes at different sites, and they each have affinity for the enzymes. And so that would be modularity. An important thing for any sort of signal is to have some way to inactivate it. So desensitization, this would be inactivation of signal. And often it's a product of the response that gets a feedback loop to shut off or remove the signal from the cell surface. And then finally, integration, when you have two signals, can have opposite effects. And so you can have a concentration of one messenger regulate the outcome of the other messenger or the other signal. And so one thing to keep in mind for an example is insulin will motivate anabolic, and glucagon is catabolic. And so there has to be a mechanism within the cell to decide, am I going to be an anabolic if insulin and glucagon are both signaling, or am I going to catabolic? And so the rule with insulin and glucagon is, of course, insulin's always going to win out. And the reason is because insulin in its signal transduction pathway has the ability to turn off messengers in the glucagon pathway. So insulin signaling will always override glucagon because it can control that pathway. Glucagon doesn't have the ability to do that on the insulin pathway. Now the first thing we're going to talk about are our ion channels. So this is a very well-known channel. It's the nicotinic acetylcholine receptor. The nicotinic choline receptor is very highly specific, so it has high specificity. Acetylcholine is the nerve signal, and this is going to trigger contraction of a muscle. So here's our muscle cell, and here's our presynaptic nerve terminal. And so we're going to have acetylcholine, which is this ACH, which is going to be released into the synaptic cleft, which is the space in between the nerve terminal and the muscle cell. And then acetylcholine released by this presynaptic nerve is going to basically bind to the receptor on the muscle cell. And this is going to cause a conformation change in the receptor, so now it becomes open. And when your muscle cell receptor opens, you're going to have an inward movement of cations. Okay, and this is going to cause depolarization of the membrane, and it's going to trigger muscle contraction. Actually, I should do arrows so you know links. So when this opens, you get movement of cations, you get depolarization of the membrane, and then you basically get muscle contraction. Now the acetylcholine receptor itself, you can see here, it's going to have 5 subunits. So we'll just do 1, 2, 3, 4, 5. So it has 5 subunits. And then when the ion channel opens, you're going to get a movement of sodium into the cell, the cation into the cell, and potassium is going to leave the cell. And then eventually you get muscle contraction that is associated with that. Now, a few of the things to think about with respect to this is that, again, we're turning on the signal through the synaptic cleft and the acetylcholine binding to your receptor on the surface of the muscle cell. And you're also going to have to turn this on. So there is acetylcholine esterase, and this is going to be when this is in the synaptic cleft, it stops the signal. And you get rapid turnoff of the signal. Now, the mechanism that acetylcholine esterase uses is not completely worked out, but it is known that the desensitization of the receptor can occur even when acetylcholine is present. So there are a lot of controls in the nicotinic acetylcholine receptor. Now, an autoimmune disease is myasthenia gravis, and this is basically where you have antibodies against the nicotinic acetylcholine receptor. And the antibodies are going to bind the receptor and then form a complex, which then gets internalized into the muscle cell. And then the muscle cell is going to degrade the messenger. And so in this particular disease, you have less muscle contractions because you're degrading the messenger for acetylcholine too quickly.